different models of DNA replication. Matthew Meselson and Franklin Stahl designed an experiment to determine the method of DNA replication. Their experiment involves the growth of E. coli on a growth medium containing heavy nitrogen. 3 models of replication were considered likely. The first is conservative replication, where the parental molecule is fully conserved and the daughter molecules are composed of newly synthesized molecules.
The second one is the semi-conservative replication. It would produce 2 DNA molecules, each of which was composed of 1/2 parental DNA, along with entirely new complementary strands. In other words, the new DNA would consist of 1 new and 1 old strand of DNA. The existing strands would serve as the complementary template for the new strand. There are 6 enzymes needed for the biochemical mechanism of the DNA replication: the topoisomerase, helicase, DNA polymerase, or what we call the haloenzymes, the primase, the ligase, and the single-strand binding proteins. The unwinding of the DNA is initiated by the topoisomerase by breaking the tension holding the helix in its coiled structure. Helicase accomplishes the unwinding of the original double strand by prying apart the double strands through breaking the hydrogen bonding affinity for each other. To maintain the stability and keep it from degrading, single-stranded binding proteins bind to it while it remains single-stranded. Then, the DNA primase lays down the initial RNA primase so that polymerase III can get to work. The primer will act as the substitute as 3-OH for beginning of the synthesizing or the replication.
After that, the main man or the DNA polymerase III will stick to this primer to build the second strand in a 5-to-3 direction. Then, the DNA polymerase I replaces the RNA in the primer with DNA. And lastly, the ligase then glues together the pieces of DNA that undergone the replication.